Okay, my name is Haim Sompolinsky, and I welcome all of you to the Brain Prize 2024 webinar series on neural circuits of the mind, frontiers of computational neuroscience. And today we will focus on a topic, sensory cognition, understanding mechanisms that link stimuli to cognition and behavior. We will have three speakers. We'll have SueYeon Chung talking about from sensation to cognition, untangling of neural manifolds in biological and artificial neural networks. We'll have Srini Turaga on simulating the brain and body of the fruit fly. Give some historical perspective on the topic that we are going to hear about in depth today. So historically, the way neuroscientists attempted to understand how the brain represents and processes the sensory environment was through the mapping of the activity of single individual neurons in the different modalities and different stages. And the classical example is neurons in the visual pathway, and the mapping started with the well-known Hartline's work in the 1930s, mapping the receptive fields of single neurons in the retina at the beginning of the visual system, and introduced the concept of receptive fields, which is essentially the patch of the visual or sensory world, in this case, a patch in the visual field for which a given neuron is sensitive to. And it's well known that he found a variety of different receptive field structures, and in particular, the one that is shown here, for instance, is center surround circular receptive field, typical for retinal receptive fields. So in this case, for ganglion cells at the output of the retina, you have center on and surround off, in this case, which means that if you have a spot of light illuminated in the center part of the receptive field, then the neuron responds, as shown here with a barrage of spikes. And if conversely, if there is a spot of light in the surround, it will suppress the activity of the neurons. Then the next breakthrough in understanding the visual processing came with Hubel and Wiesel about 2 decades later, with their groundbreaking studies of the receptive fields of neurons in the first stage of cortical visual processing, namely a primary visual cortex, or V1. As it's shown here, you can see that they found that the shape of the receptive field is no longer singular. It has a Gabor-like or elongated rectangular receptive field. And again, in this case, as in the case of the retina, at the center in this example, the neuron has an on region where the neuron will be active when there is light along the axis at the center, and conversely, it will be suppressed in its activity if the lights are the stimuli are in the surround. This led to the discovery that the neurons are sensitive primarily to orientation of the stimulus, and the notion of orientation tuning curves, mapping for each one of the neurons in that particular cortical areas. And as is well known, they came up also with a very reasonable, plausible mechanism that generates such receptive fields, by convergence of feed-forward inputs from an array of neurons with circular receptive fields onto a given simple cell in the primary visual cortex. So this is very intuitive and interesting and important way of mapping how a given population of neurons represents the stimuli simply by mapping the feature selectivity or the receptive field properties of individual neurons. Can we then, it's then very natural to ask whether we have a normative theory or optimality principle that explains the different types of receptive fields of different neurons in a given area or different areas in the brain. And in particular, in the early vision, as is well known, there are information theoretical principles, efficient coding theory, as they are called, which have a long history starting from Barlow, Laughlin, Ethick, Olshausen, and Field, Bell and Sinovsky, Simancel, and others, which, among other things, predicted or accounted for the adaptive center-surround receptive field in the retina, in the retinal ganglion cells, and the sparse coding of Gabor filters in primary visual cortex with the corresponding orientation selectivity of the underlying neurons. So this was a success story for system and theoretical neuroscience, explaining the early stages of sensory coding in the brain, and similar mapping of receptive field properties and efficient coding theories have been, similar principles have been applied both experimentally and theoretically for other modalities. Okay. So what about underlying cortical circuits? In the 90s and 2000s, decades of work proceeded in trying to understand, map experimentally and understand theoretically principles governing the local cortical circuits underlying those receptive field properties. We already mentioned the feed-forward model of Hubel and Wiesel, and later on work focused on the local cortical circuit, the ring model, the balanced state network, and the extended network models that span the entire V1 and as reasonable, plausible models of how receptive fields, properties of neurons are shaped not only by the feed-forward input, but also by the cortical feedback processing. Okay, now a large part of what we are going to discuss today has to do whether we can actually go beyond V1 or beyond that simple receptive field, individual neurons ensemble view of brain sensory processing. And that unfortunately was met with difficulty. Historically, there was an attempt to extend the principle of single neuron receptive fields and perhaps also optimality principles underlying it to higher stages. Primarily, we're talking now about the ventral stream or hierarchy of the visual cortex in the mammalian brain, particularly in primates, where the content of the images and visual stimuli were analyzed by a series of stages starting from, as shown here, from the pixelae, retinal ganglion cells, LGN, V1, which we discussed, then V2, V4, IT, etc. IT or cortex is thought to be the last stages, the last stage of purely visual processing. So the idea was that different higher stages like complex cells and stoppers, curvature cells, and so on, consist of neurons with more complex properties within the receptive field, namely receptive fields which are endowed with a selectivity to more and more complex features. This, however, was not as successful as the mapping in the early visual processing for several reasons. First of all, a large heterogeneity in the type of features that neurons were selective to. It was highly nonlinear. The whole notion of receptive fields became less powerful and predictive because neurons were sensitive not only to what's going on in their receptive field, but also to their surrounding environment. Correspondingly, computer vision also met with similar difficulties, trying to hand design features of increasing complexity didn't really lead to systems, computer vision systems with the kind of cognitive, the visual cognitive functionality which is close to what animals and humans exhibit. All right, a similar story occurred in other modalities. An example here is the somatosensory modality in the rat whisking system. Again, there is a nice and clear mapping between the whisker sensors to primary somatosensory cortex S1, and where a discrete mapping between individual whiskers and columns in S1 was found. However, if one goes beyond S1 to higher stages, one finds that neurons are sensitive to a larger set of sensors and also get feedback from other parts of the brain. So similar difficulties were observed in other modalities. And finally, at the same time, or along the recent decades, it became more and more clear that the cortical circuits is enormously complex. It may be also hold also for retina, as we may hear soon. But even S1 cortex within a given barrel or within a given column representing a single whisker, the complexity of the connectivity within that column is enormous, as has been recently shown by the connectomic mapping of that part of the brain by the Maurice Hemstadter and colleagues. So with this, we are left with the questions both about conceptually what is the proper way to describe higher level sensory processing leading to perception and cognition, and also explain them from computational principles and from underlying cortical circuitry. So I would like to end by by highlighting a problem that the perceptual system has to cope with, and we will hear more about it from SueYeon and perhaps also from Srini, that of perceptual invariance. It has been recognized in recent years that the primary goal of the sensory hierarchy is to identify high-level cognitive content of sensory stimuli or images in the visual case, which are associated with a cognitive concept or as an object, and in particular, while taking into account the fact that a given discrete cognitive concept appears in a variety of physical stimuli, physical features like changing in contrast, changing in location, changing in color, background, clutter, and so on and so forth. So it has been hypothesized that the problem of coping with perceptual invariance or generating perceptual invariance is one important challenge for sensory hierarchies. And what I would like to just end by highlighting the fact that somewhat surprisingly, even the last stages of sensory hierarchies, and in this particular, in vision particular case, the IT cortex, individual neurons are found to be still sensitive to physical variations of object stimuli, despite the fact that they are also selective to the identity of objects. So it raises the question, what is the underlying coding principle? It definitely rules out the grandmother's neuron or grandmother's cell hypothesis, because the experimental facts are strongly ruling this out as a possibility, namely that each object is represented by a single neuron. But it also rules out the concept of a neural collapse, which gained some traction in machine learning recently, namely that a given concept or given object or given cognitive entity is represented not necessarily by a single neuron, but a unique pattern of activity in the population, in this case in IT cortex, which with the property that this pattern of activity is invariant to the physical variations. That's called neural collapse. But as I said, the experimental results are strongly also opposing to this possibility. So we are with this. I'm posing to the speakers the challenge to explain to us recent advances in understanding or coming up with new approaches to how to conceptualize high-level cognitive processing of sensory stimuli in the brain and the underlying neural mechanisms for that. So with this, I would like to stop my my recording, my presentation, and I would like to are you still seeing anything?
Yes, you have to stop the share at the top of the screen.
Okay, stop share.
You'll see a button. Yeah,
okay, very good. Yeah, okay, very good. So with this introduction, I would like to introduce our first speaker, Dr. SueYeon Chung. It is a great pleasure to introduce you. SueYeon is assistant professor at the Center for Neuroscience at New York University and project leader at the Center for Computational Neuroscience at the Flatiron Institute. Dr. Chung has an exceptional academic foundation, starting with her undergraduate studies at Cornell University, where she earned her degree in physics and mathematics, graduating magna cum laude in 2009. She then went on to Harvard University, earning her master's and PhD degree in applied physics in 2017, where her doctoral work, advised by myself and Ryan Adams, focused on the statistical mechanics of neuronal representations, and in particular, understanding the concept of geometry of the representation and how it factors into cognitive processing. She then advanced her research through prestigious postdoctoral fellowships at Harvard, MIT, and Columbia, collaborating with leading neuroscientists, both experimentalists and computational, such as Jim DiCarlo, Josh McDermott, and Larry Abbott. Her pioneering work at the intersection of neuroscience and machine learning has provided groundbreaking insights into neural population geometry with application to both biological and artificial neural networks. Her contributions featured at top conferences such as NeurIPS and ICML have significantly advanced our understanding of high-level sensory representation. She has received numerous grants and awards, including the Klingstein-Simons Fellowship Award in Neuroscience. And today she will share with us insights from her pioneering research on neural manifolds in biological and AI systems. I look forward to hearing about this exciting work, and you are, SueYeon, you are welcome to share your screen and proceed with the presentation.
Thank you. Can you see my screen?
Yeah.
Looks good.
Yeah,
Looks good.
Good. Okay. Great. Yeah. Thank you so much for the kind introduction, Haim, and also a great introduction to the topic. So I'm really excited to share some of the work that my group and I have been doing with many collaborations, and focused on understanding how to think about going from sensation to cognition through the lens of neural manifolds, and using ideas from geometry and AI, such as machine learning. So the starting point of my talk is this picture where the central goal in our endeavor in understanding the brain lies in this picture of multiple levels of abstraction and multiple scales, where we have behavior and tasks and task functions and cognitions at the highest level, and individual neuron cells firing at the lowest level and the microscopic level, where the key question is to describe how this collection of neurons and synapses collectively implement behavior and tasks. And importantly, the structure of these collections of neural activities manifest themselves as neural representations. And these representations are a crucial intermediate level descriptor between cognition and task, and single neuron properties. I would like to sort of convince you at the end of this talk that's because it helps us describe and define what is the computation in the brain. And this brings us to this next question: what is a computation in the brain? There's a lot of different ways to describe it. You can think of it as input-output map, or dynamic trajectories, or a type of computer, or some kind of a learning machine with an optimization goal, and etc. But one notion that lends itself well to what is measurable in real experimental data is where we think of the neural computation as a transformation or untangling of these neural representations to implement tasks or perception or cognition or behavior. So this untangling of representations, in my view, is an important way. It gives us a language to describe the type of neural computation in the brain. And to study this kind of computation in the brain, I would like to show 3 related approaches to understand how these representations untangle for task computations in the brain, using and borrowing methods from statistical physics and machine learning theory. So first, I'll show the type of theory that can be developed for connecting the geometrical structure in neural populations, in particular describing the level of untangling of the representations and relating it to the embedded task information. And second, we're also using these geometrical insights to reason about how neural populations code information and use it as a data analysis framework for population recordings. And finally, we can combine these insights from neuroscience and theory to build better artificial neural network models of the brain. So let me start with the first part, the theory of so-called neural manifolds, which refers to the geometrical structure in neural population activities. So first, why do we care about geometry? For neural populations implementing very simple tasks, such as fine discrimination between 2 variables or estimation of some variables, historically popular approaches based on information theory or signal detection theory has worked pretty well. But then the question is, so if all of these measures work pretty well, why do you need to think about representation geometry? And it turns out most of the tasks that we face in life are pretty complex. So if you consider cognitive tasks like making a prediction about the world given a stream of video inputs, or making actions, or making inference, or navigating in a real environment, or making cognitive decisions in different types of contexts, a common thread here is that there's multiple variables in the stimuli space are at play, and these naturalistic stimuli present lots of nuisance variables in sensory space that introduce subsequently complex variabilities in the neural population activities that we, as an agent, need to be invariant to at the stage of cognition or perception. And then what do we do with these sorts of complex variabilities in neural populations? And it's really quite challenging to think about the structure in the neural activities and in relation to this kind of tasks or computations. And this is when the idea of representation geometry or neural manifolds becomes useful, because it gives us an intuition and a visual picture of how neural population activities are actually organized underlying these complex tasks. And indeed, a lot of interesting geometrical structures have been observed and discovered in the neural activity data underlying all sorts of different tasks, as I'm showing here. And now these geometrical intuitions are useful, but one remaining question is, how do we actually relate it to computation or coding? And let's be systematic about it. Let's say we're given an image or sensory stimulus, like a picture of a dog here, then a population of neurons will respond, where we can now show the spike counts or firing rate from each neuron. And that would become a point in the high-dimensional neural activity space. And this neural state space or activity space is in general high-dimensional. So for example, if you're recording from 1,000 neurons, we're talking about a 1,000-dimensional neural state space. And now in real life, this kind of an object or an image that we recognize is not really static, but rather they're moving or variable. Let's say a dog is running around and moving around, and with each of those small movement, the neurons will respond a little bit differently. And this neural representation for an image of a dog will also move around in the neural state space, and we end up getting a set of neural activities with an underlying geometrical structure, and we call them neural manifolds in neuroscience. And an important hypothesis that's gaining a momentum in systems neuroscience is if we can understand the structure and functional role of these neural manifolds, then we can learn something about cognition. But first, how do we relate the geometry of these neural manifolds with its computational property, like coding properties, or like something like a decoding performance? In other words, yes, there are geometrical structures in neural data, but how is it related to something that a brain does? And it turns out one example where we can make it tractable is what's known as invariant object recognition problem, which refers to the brain's ability to recognize and discriminate objects despite all sorts of stimuli variabilities like orientation, background, and position, and etc. And this is also a problem that a brain is solving all the time. So even now, as you recognize a word that I'm speaking, or as you recognize a face of a colleague, you're solving an invariant object recognition problem. And back to this problem of neural manifolds, now when you distinguish between two objects, let's say from dogs and dogs from cats, they will become dog manifold and a cat manifold with all those variations in the sensory space. And then we can represent the task of discriminating dogs and cats as a problem of separating these object manifolds with a hyperplane or so-called linear readout as a model of a downstream neuron that's pulling out information from that population. And the question is, can we actually connect it to the representation geometry of these activities? But we have a problem. In the higher sensory areas or cognitive brain areas where neurons are not necessarily showing simple statistical properties, in the presence of nuisance variations, you can end up with complex shapes of these object manifolds. And when you try to project those onto the decoder, you might end up with kind of a complex distribution. And it becomes very hard to analyze this with classical analysis, using simple statistical analysis. So then what do we need to do? Then what we need to do is to capture these kind of complex variabilities in high dimensional neural data in the full activity space, and instead of going into the simple decoding projection. And then measure the geometry in this full n-dimensional activity space beyond the simple second-order statistics. And the good news is there's a theory that we can actually turn to called the theory of perceptrons, which are these linear classifiers. And to account for these complex shapes in the data, we developed a new theory of perceptron on classifying these neural manifolds with underlying complex shapes. And what we have found is that the geometry of these neural manifolds is actually directly related to the system's ability to represent and recognize objects in an invariant manner. How? Before going deeper into the intuitions of what this theory gives us, let me summarize briefly what the theory is about. This capacity for linearly classifying neural manifolds, we call it manifold capacity, and that can be expressed as a function of the geometrical properties of these neural manifolds. But first, let's pause here and talk about what this capacity here really means, because it has several interpretations. First, it's about how many neural manifolds can you pack into the neural activities so that you can linearly separate them. And it's also about per neuron density-like information content about object classes. So it's about how efficient these neurons are in representing object classes invariantly in the given neural population. And then now let's talk about what this theory gives us. So just briefly, this theory of neural manifold capacity can help distinguish between inefficient, entangled neural representations, like you see in this picture, versus efficient and untangled representations in neural data. where the notion of manifold capacity captures how separable these category manifolds are, but this will be affected by the geometrical properties of neural activities, that is each of object manifolds dimensionality and radius and correlations between them. And this is actually quite similar to the problem of sphere packing in physical space, except that we're talking about neural object manifolds in the neural activity space. So for example, if these object manifolds and neural data are high dimensional and large, then you can only represent small number of these object manifolds, and vice versa. If they're small and low dimensional, then you can represent many of them such that you can linearly classify them. This is just like how if you have smaller balls, you can pack more of them in a box, or if you have disks, you can pack more of them than having three-dimensional balls in the same volume of the box. In terms of the locations of these neural manifolds, if they are spread out, you can pack and represent more of them. And in terms of their directions, if they're more aligned, you can pack and represent more categories in the brain, just like how if you can stack coins together, you can pack more rather than compared to packing them randomly. And you can see that this kind of framework connects the also different levels of abstraction and scales, where the manifold capacity talks about how many object categories you can read out, which is more related to cognitive performance or capacity, while these geometrical measures are more related to the neural responses in response to the. Now, this theory can be used as measures for the goodness of neural representations in the brain. And just to quickly summarize the theory, so this can be used as a multi-level probe for neural population, where given the neural activities with categories, by the way, they don't have to be smooth, meaning that you can have point clouds with underlying structure. This method and theory will compute the geometrical properties and predict the manifold classification capacity. And this has interesting implications for both neuroscience and neural network studies. Let me start with the implications in neuroscience. First of all, this method doesn't rely on dimensionality reduction idea like many of the geometrical visualization methods assume. And you don't really need to throw away most of your data in order to visualize it in 2D or 3 dimension. And instead, it measures these geometrical properties of neural activity directly in the high dimensional neural activity space. And secondly, this theory can give metrics or measures at 2 levels of abstraction, and while providing an analytic link between the task level efficiency at the cognitive level and the neural population structure again, from sensory representations. And from the perspective of the theory of neural computation, this kind of neural manifold can provide mechanistic underpinning for implementing a task and generate the population level hypotheses and scenarios, because even under the same level of task capacity or behavioral performance, you can have multiple different geometrical configurations that will implement those. And it's also exciting because this kind of neural manifold geometry is a consequence of collective properties of single neurons, such as tuning curve structure or their distributions. So by connecting the population level geometry with the task level performance, we're one step closer to understand the role of individual neurons in task implementation. So that was about the theory, but let's talk about the application of this theory, particularly as a data analysis framework, and to characterize how different brain regions transform these representations from early sensory areas to higher sensory and more cognitive brain areas. So let's look at how you can analyze the real data, first from features of deep networks as a test bed for neural recordings, which is coming in a few slides. But here we run these images of objects with different classes or categories and show this on AlexNet, which is a well-known visual deep network. And you can extract features similar to the real neural population data. And from these features, you can recover these object manifolds. Then we're ready to apply this neural manifold capacity theory. So first, this is a sanity check that the theory works, that the predictions from the theory compute the manifold capacity that's pretty similar to the empirically measured manifold capacity. Then if you look at the measures across the layers of this deep network, and then follow this manifold capacity measure and then 3 geometrical parameters across each one of those layers, what we see is that the measures sort of move as predicted, but gives us a concrete number that give us a sense of how much information is in that representation. So for example, the object manifold capacity for those object classes and images, they increase across the layers as they transform across the layers of a deep network. And the increase in the object manifold capacity is because there is a reduction in the dimensionality and radius and correlations in these between these manifolds. And what this is telling us is that these object manifolds are becoming not only more separable, but also more efficient in terms of being able to represent more of them in the deeper higher sensory areas that are closer to the cognitive perception, like environment object recognition. But how about the biological neural data and recordings from real animals? So this was the first use case from macaque monkey's visual cortex, especially from the brain area called V4 and IT, the areas that are responsible for object recognition, because if you lesion this area, the performance of object recognition suffers in the animals. So we took the neural activities from hundreds of neurons recorded simultaneously from these brain regions, while the monkey is seeing many of these different objects, images. And in the data, we can identify these object manifolds and characterize the manifold capacity, which increases across from the pixel level representation to V4 to IT, and characterize a geometry and packability of these object manifolds in the neural activity space. And as you can see, the capacity improves as we travel downstream of the visual hierarchy in the ventral stream. And we can also see that that's because the structures of these object manifolds are transforming such that the dimensionality is getting smaller, so they're getting flatter in the activity space. The radius is getting smaller, and they're getting also smaller, and so on. So with this kind of a new analytical theory, we can get a nice coherent trend across the visual cortex hierarchy, despite the fact that at the individual neuron level, they show complex shapes. And since then, we have been using this kind of geometrical framework to study a lot of different problems in both neuroscience and deep learning, going from vision to audition to language problems. And we also have a growing list of experimental neural data sets that we're helping to analyze, and the species can go from monkeys to humans, and across different modalities. So that was about neural manifold theory as a data analysis tool, but can we actually use these theoretical and experimental neuroscience insights as a design principle for better network models for the brain? And that brings us to this last part. So as Haim introduced, in vision, it has been long assumed that the sensory neurons are adapted through both evolutionary and developmental processes to the statistical properties of the signals to which they are exposed. And then this goes back to many of the names that were already mentioned in the introduction. And these ideas that connect the information theory to provide a link between the environmental statistics and neural responses through the concept of coding efficiency have turned into methods for connecting statistical models for visual images and ways to validate these models for single neurons and their properties. And this kind of efficient coding hypothesis has become experimentally testable for both individual neurons and populations of neurons. And it was a hugely successful endeavor for a model of early sensory processing stages. But using these ideas for reasoning about downstream visual areas has been a challenge. And one reason for that is that this efficient coding for early vision was mostly focused on preserving as much information about the images in the neurons, while for late visual areas, you need to think about how to transform these neural representations and actually throw away some of the irrelevant information so that these neural representations become efficient for downstream tasks when the tasks need to have some sort of invariance. And this is where the idea of the manifold come in. We know that the manifold capacity measures how packable these neural manifolds are in the neural representation space. But if you think about it, isn't it actually a form of a task efficiency? So if that's the case, can't we actually train a deep network with it? So this is exactly what we did. So we used the standard deep neural network model called ResNet as a backbone and generated these neural manifolds by introducing small variations of input images. It's known as data augmentation in machine learning, and compressed these image variation manifolds while expanding the size of the overall global manifold across images to maximize the packing efficiency of these neural representation manifolds. In other words, we tried to maximize the manifold capacity in the representation space of a deep network. Then, even though we didn't use the labels for object classes, it turns out that this kind of paradigm learns object manifold representations with the performance that's actually quite close to state of the art for invariant object recognition performance, and also it does quite well on transferring to other tasks. And perhaps more strikingly, it turns out that it can also, the features from these models can predict neural activities from middle layers of ventral stream in monkeys' visual cortex pretty well. So what we learned from this is that learning the efficient and dense packing of image variations turn out to be a pretty good learning objective for learning how to recognize objects without using category labels, and also predicting recordings from middle layers of ventral stream in monkeys. And in other words, using the theory of neural manifolds can help design better deep networks, both for the task performance perspective for applications, but also for the brain model perspective. So I hope I was able to convince you that the theory of neural manifolds can be viewed as a new theory for neural coding in populations of neurons and new data analysis framework for highly structured data in populations of neurons, and also a design principle for better deep network models. And I think it's exciting because it formally connects the multiple levels of abstraction and scales, because the manifold capacity is a task or cognitive level measure, while the representation geometry is a collective neural activities and their structure. And it also captures how efficiently information is packed in neural populations, and it generalizes this notion of efficient coding to task level efficiency. This method is applicable to a wide array of data sets and super versatile. And it also helps build and interpret deep learning models, neuro-inspired or otherwise. And perhaps most exciting aspect for me as a theorist is that this kind of framework is extendable to many other tasks beyond object recognition and categorization. So I think this is just the beginning of many of the exciting future work that can be done. And with that, here are the references for the work mentioned during the talk, but these are the works that are highlighted in the talk. And I hope I was able to convince you that by using analytical methods from statistical physics and machine learning, we can build the bridges between multiple scales and levels of abstraction and build a normative theory of representations and use it as a way to both build a model of the brain, but also as a data analysis tool. And with that, thank you for your attention, and thanks for having me.
Okay, and thank you, SueYeon, for the fascinating talk. Before we move on, Srini, do you have any short question, kind of technical question for SueYeon, because we'll have a broader theme discussion at the end. Go ahead. You have any question?
Yeah, SueYeon, great talk. That was cool. I think I see where this is coming from the perspective of categorizing objects when the stimulus falls into categories. I'm kind of curious if there's extensions to, imagine that I want to pick up a cup first. I want to know where the cup is and that there is a cup, but also want to encode certain things about that cup. Like where would be the handle, where can I hold it, which way is it pointing, or if I want to catch a ball, where it's coming from, perhaps features that are more continuous valued. I'm kind of curious if there's any bridge from where you are to the decoding of continuous variables as opposed to categorical variables.
Yeah, so that's one of the directions that I'm very excited about, and the short answer is that if you zoom out and then use the similar kind of mathematical approach or a stat physics approach, you can build a theory that is about decoding continuous variables in the presence of invariance, and maybe you can ask me in a couple of months, and then I can tell you more about it.
Okay, I have, thank you. I have a brief technical question about the self-supervised algorithm that you mentioned. Don't you need to give during training the signal of identity of several images that they belong to the same category in order to implement it? Or is it completely label-free?
It's completely label-free. During the training, it doesn't have access to any of the object classes.
No, I know, but does it have access to the fact that two images belong to the same category or not?
It doesn't.
It does not. I see.
So yeah, the manifold from this perspective is actually image variation. So every image forms a small manifold.
Yeah. I see. Okay. So you just look at a given image and introduce variation to it. Okay.
Right.
Okay, very good. Okay, that's very exciting. Okay, so let me introduce our next speaker. It's a pleasure to introduce Dr. Srini Turaga, a group leader at the Harvard Hughes Medical Institute, Janelia Research Campus, where he has led groundbreaking research since 2014. Srini obtained his PhD in computer science from MIT under the supervision of Sebastian Song, making significant contribution to the field of machine learning and neuroscience, particularly in reconstruction of neural circuit connectivity using 3D electron microscopy. A key highlight of his doctoral work included his collaboration with Winfried Denk, a Brain Prize winner, on the mapping of the circuitry of the mouse retina, a project that laid the foundation for the crowdsourcing platform EyeWire, led by Sebastian Yu. So, following his PhD, Dr. Turaga pursued postdoctoral research at the Gatsby unit at the University College London, working with Peter Dayan and Michael Hauser to develop statistical models of large-scale neuronal recordings. At Janelia, Srini leads cutting-edge projects focused on mapping of neural connectivity and modeling the associated neural activity. His recent efforts in modeling the fruit fly brain represent a bold attempt to bridge the gap between the static connectomes and the dynamic computations of the living brain. We are excited to hear more about this visionary work in simulating the entire pathway from stimuli to brain to body in the fruit fly. Let's welcome Dr. Turaga to share his insights with us. Please.
Thank you, Haim. SueYeon, I'm going to share my screen, but I might ask you to unshare. Did it work?
Yeah.
Excellent. Great. Well, thank you. Thank you for having me. It's a wonderful opportunity to present our work. I'll start by acknowledging the heroes of this work. The bulk of the work that I'll present was led by PhD student Janne Lappalainen, who's jointly supervised by myself and my close collaborator Jakob Mage. And I'll also highlight some work by other people listed here. So the fruit fly, which I've become familiar with since my time here at Janelia, is a tiny insect, 2.5 millimeters large, has a short lifespan. The very exciting thing about the fruit fly is that it has a tiny brain, and with this tiny brain, it does a lot. It can look for food, look for mates, court mates, fight off other fruit flies, and escape predators. So it does all of this with a brain that's just about 160,000 neurons. And super excitingly, in just the last year, we now have a connectome for the entire brain and the entire ventral nerve cord of the fruit fly. A connectome is a map that tells you the wiring of each of these 160,000 neurons and who it's connected to and how strongly they're connected in this whole brain. And the other nice thing about the fly as a model organism is that its wiring diagram is largely conserved from animal to animal. And so to the point where you can find the same neuron and these neurons have names. And so you can make a recording of a neuron in one animal, you can look at the connectome in a different animal, and we can, they'll largely have good correspondence. And so this has been used by the community for quite some time now in making measurements across the fly nervous system and then putting them together into a common understanding. Now, my job is to try to take this connectome and to build models from it of how the living brain functions. The connectome comes from a dead brain. And so let me give you my caricature as a computational neuroscientist of how I think the brain works. At the microscopic scale, the brain is a neural network. And so as someone who's been, comes from also the AI and deep learning world, we know that it matters how that network of neurons is connected. What does the weight matrix look like, for instance? From the neuroscience perspective, we know that different neurons can have very different biophysics, which determine the input-output function of a neuron. And similarly, synapses themselves can have very interesting and exciting biophysics, which control the input-output functions of those synapses. And so that means that when you assemble these neurons together into a network, all of these details at the microscopic scale matter. And further, there's a host of other phenomena like neuromodulation, plasticity, and perhaps more that we have yet to discover that together collectively give rise to the neural activity. Neural activity isn't just a function of a single neuron, it's a function of the network. And the neural activity produced in the nervous system drives the behavior that the animal generates. And the behavior usually is in the service of some task or goal at the cognitive scale that the fly or animal has set for itself. Now, super excitingly, we now can measure the connectivity. But we can only measure the connectivity and comprehensiveness right now. We can't get all of these other measurements at the same time in sufficient sort of comprehensiveness. And so this has led people, about 20 years ago when I first got started in my PhD and working in connectomics, to critique the idea of assembling these connectomes, which come at great cost, because connectomes by themselves are not sufficient, we know, to explain neural activity, behavior, and how the brain works. It gives us a lot of detail, but it doesn't give us everything because it's missing the dynamics. The input-output functions of neurons and synapses, for instance, are just one component of measurements that you can only make in the living brain. And the connectivity is made in a dead brain. And so, how do we bridge this gap and how do we go from these measurements that we now have to an understanding of the whole brain? Now, the good thing, of course, is that we can measure a lot of other things from the living brain. We can measure behavior. We've been able to measure animal behavior for a long time just by looking at it with an eyeball. Now we can do it with high-speed video cameras. We've of course been able to measure neural activity and dynamic biophysics, etc., in living brains, but it's usually not comprehensive, and it's usually one neuron here, one neuron there for the biophysics, for neural activity. Even if you can measure whole brain neural activity, it's usually under very special circumstances, so not under free behavior. And so you can a lot of these things you can measure, but not all the time. And so the question that we had set to ourselves is, can these overlapping measurements that we can now make from the nervous system help us build up the full picture? In particular, what we asked was, imagine that we have the connectome of a neural circuit, which we get from a dead brain, and we know something at the cognitive level about the task, the computational task that a circuit is trying to perform. Is that actually enough to bridge these scales? So if we have measurements or some understanding of the circuit at these two, at the very microscopic scale and at the macroscopic scale, can we predict neural activity in the living brain? And so that was a thought experiment. And the reason we sort of started with this is that we kind of know from computer science that if we know the single neuron transfer function, but don't know the connectivity, that there's a very large space of solutions that we can't uniquely identify neural activity. But the converse is the converse truth. We know connectivity, but we don't know the transfer functions, the single neuron activation functions, for instance. Is that sufficient? Does that really get us? Is that really the most important measurement that we can make? And can we get from this dead brain to a living brain? We studied this question in the fly visual system, where there's been a huge amount of research mapping the connectivity since kind of the beginning of Janelia here. Janelia is one of the places where connectomics has really been pushed forward, especially in the fruit fly nervous system. And the fly visual system was where we had the most data. And so we asked, if we would take these connectomic measurements in the fly visual system, can we build a simulation that can then make predictions of neural activity which we can then compare to measurements of neural activity made in the living brain by folks across the community. In this way, without ever having used a single neural activity measurement to build a model, can we now generate hypotheses about how the brain actually works? And if we can do that, that means that this measurement of the connectome is a really significant and important measurement to have made. So I'll give you a very quick overview of the anatomy of the fly optic lobes. The fly has two large compound eyes. They dominate both the head as well as its nervous system. Each optic lobe accounts for maybe about a third of the neurons in the brain, and together they're 2/3 of the nervous system. So it's a very highly visual animal. And further, these two compound eyes have photoreceptors organized in a perfect crystalline hexagonal lattice. That's beautiful. But the especially beautiful thing for us is that so is the neural circuitry that processes the inputs from these photoreceptors. The neurons themselves also have this crystalline hexagonal lattice organization in the lamina, the medulla, the lobula, lobule plate, which are different stages of processing within the optic lobe. Now, when we worked on this project, we had the connectivity for just about 64 cell types in the fly visual system. And this is the matrix that you see on the right, is the connectome for those 64 cell types. And it just tells you which types are connected to each other. You'll note first that it's highly sparse. And then you'll note that there's these squares that have different sizes, and some are blue and some are red. So from transcriptomic analysis, we also know whether synapses, particular synapses between cell types, are excitatory or inhibitory. And that's what's indicated by red and blue. We also can count the number of synapses between any two neurons of cell type A or cell type B. And that tells you, and the size of the square tells you how strongly they're connected. And the key is here on the right. And you'll note that pairs of neurons can really have lots of synapses between them, and there's a large dynamic range there. And what we've discovered in the fruit fly is that the number of synapses between any two neurons is a very good proxy for the strength of a connection between them. It has a very large dynamic range compared to the sizes of individual synapses. And so it's very likely, and a hypothesis in this work, that the number of synapses is the main dominant factor of how strong a connection is between two neurons. Now, we don't just know the connectivity between cell types, we actually also know the connectivity at the level of individual neurons. And that's what I'm showing you here in this matrix on the right for a local patch of the optic lobe, because I can't show you all the neurons. You wouldn't see anything. And what we know is for, across the retina, for each neuron of a cell type, and neurons of any other partner cell type, what the spatial organization of the connectivity is. So as I told you, these neurons are laid out in a hexagonal lattice where there's one neuron per column, and each column is responsible, you can think of as for a pixel of visual space. And so what you're seeing on the left here is the spatial organization of inputs that this neuron, this T4D neuron, as I told you before, fly neurons have names. And so you'll see lots of these names that people know a lot about. But for the most part in this talk, I'll try not to focus too much on these names. And so this is a highly studied neuron. It receives input from this other highly studied neuron called MI9 in this spatial organization. And so there's actually a strong connection between the kinds of modeling that we've done and what SueYeon's talked about and what Haim talked about at the very beginning. We can think of all of these, our model is out of a convolutional neural network, but in this case, a recurrent convolutional neural network, where what you see here are each hexagon is a particular cell type, one of the 64 cell types that I've talked about, for which we know the connectivity. There's edges between the hexagons if there's connections between those cell types. And further, each pixel inside one of these hexagons corresponds to one of those neurons responsible for that part of visual space. In reality, these neurons are all tangled up, but in our model, we can kind of tease them apart and look at each, all the neurons of a particular cell type together. And the correspondence between this model and convolutional neural networks is that you can think of each of these cell types as a feature map in a convolutional neural network. And the edges between them correspond to the filters between those feature maps. Now, there's feed-forward connections as well as feedback connections. This is, if you look at it, there's a lot of sort of a mess of tangled connections here, but I assure you it's still quite sparse. Now, the goal of our exercise is to be able to predict, given a video clip presented to the retina, so a dynamic visual stimuli, what would the time series of neural activity look like across the entire model that we've got here? And we're modeling about 64 cell types, which together comprise about 70% of the optic lobe. So this together is a very large fraction of the entire nervous system that we're modeling, and a huge number of cell types here. So it's a fairly detailed model that we'd like to be able to build, and that we have the measurements for to be able to build. So this model has a one-to-one correspondence with the living brain. Every neuron and synapse in the model corresponds to a real neuron and synapse in the living brain. So now the question is, how can we simulate this? Because the thing that's missing is we don't know the dynamics of these neurons and synapses. So that's the piece that we need to know in order to be able to bring this network to life. So what we decided was for this model, we wanted to focus on having detailed connectivity, which is what we have direct measurements of, but simple models of the neurons. So the neuron models themselves are not complicated. You can really spend a lot of time and effort modeling the great deal of complexity, all the ion channels, conductances, and so on. But we wanted to see how far can you get with just very simple neurons. And so the only equations slide that I have here, I'll just read off the parts on the left because those are the most important ways to think about this system. Each neuron is modeled as a point neuron, a passive point neuron. Even this model has two parameters for each neuron. One is a time constant, which says how quickly does it respond to inputs. And then the second is the resting membrane potential, which says, in the absence of input, what's the voltage that neuron sits at. So those are things we don't have direct measurements of. Now we have to figure out what to do. The thing that we do have measurements of, mostly, is the weights that connect these neurons. And so the weights that we have here, the Wij that I show you over here, has two components, three components to it. One is the number of synapses between any pair of neurons, and that comes from the connectome. We have a direct measurement of that. The second is the sign of a neuron, of a synapse. This also comes to us from transcriptome. So the connectome and the transcriptome together give us N as well as T, sigma, between any two neurons. What we don't know is how much current does one of those synapses convey. We don't know how strong a single synapse is between any pair of cell types, and it's determined by a lot of biophysical parameters. It could be how many how quickly does it release neurotransmitter, how many receptors are on the postsynaptic side. These are things we don't have direct measurements for. And so that's another free parameter in our model. So we have really three kinds of free parameters in our model. We have the single neuron parameters, the time constant and the resting membrane potential, and we have this unitary synaptic strength. Now, what we said for this model is whatever those parameters are, they're probably the same for neurons of the same cell type. And so that brings us a massive compression in number of parameters, down to just the number. It just scales with the number of cell types and not the number of neurons. And so that's going to be one time constant per cell type for all the neurons of that cell type. Now, if we didn't have the connectome and we didn't have cell types, we'd probably need about 2 million parameters to really be able to explain, to fit a model of this complexity. But with the connectome, because most of the parameters are actually in the weights, and because we have those weights, direct measurements of those weights, we really only have 700 free parameters. It's still a lot. I can't just sort of twiddle with it one number at a time and get to everything. And so this is where, our hypothesis that if we know something at the cognitive scale about what the circuit is doing, that we can sort of, this is where that comes in. And so here's what we said. Our connectome that we have is largely for the motion pathways of the fly visual system. And that's something where there's been a huge amount of research in the fly visual neuroscience world. And so this is an area that's super important to a fly. Flies care about motion so that they don't get eaten by predators. They're highly sensitive to motion in their visual environment. And so we said somewhere in this network, it needs to be able to detect motion. And so what if we optimized those missing parameters that we don't actually have measurements for, the dynamical parameters, until this network as a whole, somewhere, is able to detect motion. So the way we did this was we took some computer vision databases of optic flow. Optic flow is how computer vision folks think about motion, where you have a video clip, a video has frames in it, and for each frame and for each pixel in that frame, you can say, where does that pixel go in the next frame? And so that's a vector that connects the pixel's current location to the future location in the next frame. And you can make a vector field like that, and that's what you see here on the right. The goal is to then predict that vector field from this video clip of many frames. And if we can do that somehow, then that means that somewhere in this network, the fly is able to detect motion. So the way we did this, we set up a motion decoder to basically transform the coordinates. Essentially, somewhere in this network, the fly is detecting motion. We don't know where, which neurons, and how. And we want to transform that to the computer scientist representation of optic flow. That's what the optic motion decoder is doing. And then we have this system of coupled differential equations for about 45,000 neurons here that we're simulating that have these 700 free parameters. And we simulate them and then optimize using backpropagation through time until we can find parameters that are able to do the optic flow task as best as this tiny fly simulation can do. So once we've done this, we now have a simulation that we can show any video clip to and generate neural activity across this entire circuit. And these are predictions because we never ever used real measurements of neural activity to build this model. And so what we can do is we can go back and look at 20 plus years of research that fly visual neuroscientists have done in the optic lobe, recording from various cell types. And they've really only recorded, even up to now, maybe only about half of those cell types in this that we are modeling. But we can go and look at the laboratory stimuli that they have used and simulate them and ask, how does our neural activity match? Now, one of the famous neurons I mentioned is a T4 neuron. And the T4 neuron is cool because it has been shown to respond to motion in this very particular way. So if you have edges that are moving upwards versus moving downwards, this neuron will respond more strongly, this T4C neuron, to one direction, which we call the preferred direction, as opposed to the opposite direction, which is the null direction. And so what you're seeing here is the predictions of our model for the motion in the preferred direction versus the null direction. And the fact that we see this contrast means that this neuron is motion selective. And so that's super cool because that was a fundamental foundational discovery made about 10 years ago by Axel Borst's lab. In fact, here's what their data looks like, the Mysak et al. For the 4 subtypes of T4 and T5, which have been shown to be motion selective in the 4 cardinal directions, and black is their measurements from that landmark paper. And we're able to, using our model, pinpoint exactly which neurons are motion selective and show that they are tuned in these 4 cardinal directions and make that prediction accurately. So that was super cool. In fact, you can go and look at the motion selectivity indices across these 64 cell types that we're looking at, and we correctly predict that only those neurons are motion selective to their well-known preferred stimuli and not the input neurons. And then we also have some extra predictions for other neurons which might be motion tuned, which have not yet been characterized experimentally. So these are extra predictions. And really, if you look at the circuit, about half the cell types have yet to be characterized. The simplest characterization that's been done is this well-known characterization into segregation into on and off pathways that Haim talked about at the beginning. And we know for at least half of them, whether they're on or off, and our model predicts them perfectly well. Each circle here is actually one model, and a model ensemble trained in this way. So we trained 50 models to do the same task with exactly the same connectome, but we found that they have different solutions. So this is an interesting and useful way of characterizing the space of solutions that are consistent with having to compute optic flow and have the same connectome. And so one question that we asked, and then going on, you can also look at really mechanistic predictions on what's happening in the circuit. You can ask, what are the excitatory and inhibitory components that give rise to this? This is something that fly motion aficionados have studied and debated for a long time. Our model can make these predictions, and these actually are in good agreement with experimental measurements as well. This is at the level of excitatory and inhibitory currents. Further, we can actually break it down by input. Which input is coming from which cell type. This is a measurement that we can't even make right now. So that's the power of the model is sort of going beyond the measurements that are possible. The last thing I'll just say is that this model is actually a model ensemble. It generates multiple predictions, 50 if we train 50 models. So you can ask, does it make 50 predictions? Are they different predictions? Or how well constrained are the predictions made by this model? And it turns out for T4 at least, they're very well clustered into 3 clusters. And these clusters actually correspond to interesting mechanisms. If you look at the top 2 clusters, they correspond to these input neurons having different contrast selectivities. And so what this means at a high level is that with a model like this, before you had a model like this, if you looked at a particular cell type and you asked what would its tuning be, you had no idea at all. Now we can give you a small number of predictions. And further, what we can tell you is that if you make this one recording from this one cell type, that will tell us about all the other cell types that it's connected to as well. And that's power that we only have through the connectome. And so I think this modeling framework can serve as a hypothesis generation and discovery tool for not only generating hypotheses, but optimal experimental design. So we have, at this point, we put, I think, something like 650 pages of predictions, because for each cell type here, we have a huge number of predictions in this mechanistic model that we have as a supplement to this paper. I hope I've convinced you that through this, that connectivity, the connectome, is super important measurement to have made. Now that we have it, we can really go far in predicting neural activity. And that wouldn't have been possible with other techniques. And it's only possible because we have this connectome. If we had trained a black box neural network to perform the same task, we wouldn't have been able to make these predictions at this resolution, at single neuron resolution, at single synapse resolution even, predicting the roles of individual neurons and synapses in performing a particular cognitive task. Now, in just maybe 1 or 2 minutes, I'd like to summarize with a vision for what we can do now with this big connectome that's just sort of landed on our doorstep. We now have the connectome for the whole thing, the whole nervous system, and there's more coming. What can we do here? Well, the thing that we showed is that if you know the input-output function for the circuit and you know the connectome, then you can make the predictions at the level of single neurons. So what's the input-output function at the level of the entire nervous system? Well, the inputs are the sensory system and the outputs are the motor system. And they communicate with the world through a body, and the body is how it interacts with the environment. And so if you have measurements of behavior and you have a model of the environment and the body, then you can infer on a moment-to-moment basis what the sensory system is receiving as input and what the motor system is producing as output. And so what we did was we built that body model. We measured the body in great detail using light microscopy. We constructed the geometry of the body, and then we gave it life inside of a physics simulator called Mujoko. And we then trained this model to imitate real locomotion using real measurements of flight and walking, made in the labs of Christian Branson for walking and Michael Dickinson for flight. And what we've shown is that this model now walks and flies in the same way that the real fruit fly does. And so this gives us a bag of techniques, I think, now, the model components as well as techniques, that might help us dream bigger than we have before, and maybe even dream big enough to build a model of the entire fly nervous system, because we now have measurements of the connectivity at the level of the entire fly nervous system. So with that, I'll thank you and take questions.
Thank you very much, Srini, for this very exciting presentation of your work. Again, before we start a brief discussion, SueYeon, do you have a technical question to Srini?
Yeah, I was wondering in the second, in the last few slides of this virtual, the model of the fly, I was wondering what kind of objective function went in, if there's something that we can learn about the type of computation that the fly is optimizing for. Is it like an estimation of where it should go, or prediction of what will happen, or anything?
That's a great question. And so what we trained here is a closed loop controller that will walk in closed loop or fly in closed loop the same way that these flies walk. And so we had video clips of fly behavior, and we said, how do you learn to match this? And so we call it imitation learning. And so it's a form of reinforcement learning that we used. And so what that means is, the body model, our simulation, is feeding it the sensory inputs on a moment-to-moment basis, and is saying, you need to generate the same motor output in order to be able to generate that exact trajectory of flight or, walking. So that's sort of the objective function.
Was it supervised with the action?
Not with the action, but the result of the action, the positions.
Right.
Yeah. Because the action itself is yeah.
Yeah, thank you. I have a question, a technical question about the ensemble of models that you find, ensemble of solutions, and you said I think there were kind of 3 clusters of solutions. It's intriguing because I don't know of similar phenomena in deep networks where if you look at the ensemble of solutions that one get, let's say for deep convolutional networks in vision, I'm not familiar with this kind of sharp clustering phenomena. I wonder if you can say you can comment on it. In particular, can we would you speculate that different hello? Yeah. Would you speculate that different clusters perhaps correspond to perhaps different individual flies sharing the same connectome?
Yes. So I'll start with a bit of intuition about that I have about what we're doing. What we're doing is we're not training the weights. We have the weights, they're given to us, and the weights are so sparse that my hope was always that there would be so few ways of actually computing the input-output function that we know that the fly can do, that might allow us to identify, to make the hidden layers, the hidden units identifiable. And so, and what we show is that that's actually indeed the case. And so that's the way in which it's different from the training of artificial neural networks, where you're actually training the weights. Here, the weights are actually given, and all you're training is the nonlinearities, so to speak. And so that's a far tighter, and especially because it's such a sparse neural network, that can probably, the representational capacity of the connectome constrained network is likely to be extremely small. And really targeted towards the functions that it needs to do. And so that's what gives us the hope of cracking the code in this way.
And would you speculate about the possible variation between different individuals?
Yes, good question. The variations that we've seen so far correspond to unknowns in the circuit itself. Like there's these T4 neurons have flanking inhibition, one on the left, one on the right, and depending on which neuron is on versus off, that can change basically whether it's more selective to motion in one direction versus the other. So that's a particular degeneracy that exists here. And it's unlikely, to my mind, that that would be an individual variability. That the transcriptome and yeah.
I see. Okay. So if the last question, which actually will bring us back to the broader issues, is I'm trying to think: is there a way to connect the two approaches, SueYeon and use, and in particular, can you use connectome or the methodology that you described to to tackle more high-level questions about cognition. For instance, I'm sure the fly has also kind of perceptual invariance tasks and so on. So do you see a way to connect the two approaches?
SueYeon, do you want to go first, or shall I go first? Who's it for, Haim?
No, well, I see it as a follow-up questions on your talk. So then SueYeon can go. So first, let's hear you.
Yes. So I actually, after SueYeon's talk, made some notes to myself that basically said, look at the manifold, the capacity as we go through the fly, different stages of the what does the representation look like? We should start to characterize these things. We haven't yet. I think that would be amazing. And then the other thing I thought of is, can we use this unsupervised framework of MMCR to train this model in a task agnostic way? And this is where some of my questions to you, SueYeon, came from, because the fly is probably trying to also compute, not just categorize the world, but also estimate distances, and will this looming object come hit me, or will it miss me? And so I think all of these, we don't know. We don't have yet the full repertoire of all the tasks that the fly visual system was evolved to perform. We started with one of these, which is optic flow, but that's clearly not the only thing it does. It does many other things, and some of this degeneracy may even go away if we knew the full selection of things that it's actually designed to have solved. But maybe some of these other techniques, like your SueYeon's unsupervised learning, could help us sort of crack this code enough without further supervision. So those are two things that occurred to me that we should work on, and it would be great to work with you on this, of course.
Yeah. Okay. That's the webinar may be a catalyst for this type of collaboration. I look forward to it. So, but SueYeon, do you have a comment on this particular question? Can you see your methods applicable to fly?
Yeah. So there's two variations of the theory that comments. So one is on the MMCR. We actually have a paper that's forthcoming in this year's NeurIPS on introducing equivariance, which is not, while trying to have all this efficiency, but you try to preserve the encoding of these continuous variables. If you introduce that into the objective function of the network, it actually, the features end up becoming more similar to the neural recordings in the monkey vision. So it is possible to extend the MMCR framework, or more broadly, self-supervised learning objectives, to include the sensitivity to these continuous non-categorical variables. So it'll be great to actually find a way to combine these approaches. And the other comment I have is, or I guess it's more of a question to Srini. If I was wondering if it's possible to have additional variables to capture this sort of a mixed selectivity kind of aspect, or invariance into your stimuli or description of a behavior, so that you can partition the data or recordings or representations in many interesting different ways. And the other question, the question 2, is whether your approach might end up leading to new modules or network motifs that can be broadly applicable in machine learning models. Just like how receptive fields or statistics of receptive fields can be used as filter banks, and maybe here you can have cell types or the shapes of responses given different between different cell types. So yes, this is more. My question is probably much simpler than your answers.
No, I wish that were the case. I think we have yet to look in this model for what principles we can extract from this. We've been focused largely on building as faithful a model as we can, faithful to biology, and focused on mechanistic insight about what does cell type A do, what's the role of this particular synapse in the system, but not yet, and woefully, insufficient amount of time thinking about what normative principles can we learn from this, because the fly is clearly doing a lot with very little, and it's an incredibly efficient system. So that there's got to be a ton of things we can learn from this, and I'm embarrassed to say that we have yet to look at this question.
Okay, so I would like to go back to SueYeon's presentation and ask you.
So I'll stop sharing, Haim. Is that good? And we'll have a discussion.
What?
I'll stop sharing.
Yeah, you can stop sharing. Yeah. Yes. Great. Thanks. SueYeon, I have several questions that you may be able to elaborate. First of all, you emphasized rightly the commonality between the way deep artificial neural networks process sensory information or represent sensory information, and the way the brain does. But have you also found substantial differences between the two systems? And if so, can you tell us what are the conclusions from this?
Yeah, so using the geometrical approach to distinguish between similarities versus differences between the brain activities and the features from the network, and also among the models, this is at the infancy. It's really, we're kind of at the early stage of starting to look into these sorts of questions, but I think there are many interesting early findings that we're starting to see, such as different types of objective functions. So depending on what kind of task or computation was used to train a network, the resulting geometries can differ. So for example, between the supervised learning versus unsupervised or self-supervised learning, the types of geometries measured with these manifold geometrical measures can be quite different. As you can imagine, if you use unsupervised or self-supervised objectives, you end up with representations that are more general purpose good representations than geometry that's specialized for a particular task that's been trained with such a task. But I think that's probably one of the things that you can do, but there's many other things that one can go after to distinguish between models or between the brain and the model. And I think it's one of the strengths of this kind of approach, that you can have access to the geometrical measures beyond just the similarity.
Yeah, but I guess the question is, even within the same objective function, for instance, if you do somewhat similar to what Srini has shown, if you run the model with many different initial conditions, mm-hmm. and you compare the representational geometry, are they all similar, or there is from the perspective of the geometry, you cannot really distinguish in any meaningful way? Or you do expect to see some differences? Right.
So this is I think some of my answer here will be very speculative, but I guess there's the one actually interesting question that might be related to Srini's approach. even when you have the same, everything else be the same, depending on the strength of the weights that you set at the initial condition, for example, people have reported that the resulting learning dynamics is quite different. This is known as lazy versus rich learning. And we found with our method that the resulting geometries between these different regimes are different. So I guess maybe that can be a sort of like a kind of loose prediction on that can be translated to connectivity measures, perhaps. Yeah, depending on what was the initial strength of the synaptic connectivities. Yeah.
Could I jump in on a slightly complementary?
Sure.
So I think SueYeon, the framework that you presented certainly had a task component to it, and in performing a task, you don't really need to encode everything about the stimulus anymore. Sure. You can start to throw away information as you go through, and so some of what's happening is not just the rearrangement of information inside these manifolds, but also throwing away of information that doesn't have to do with the task. I'm kind of curious now in our work. One of the things we tried based on the work that Haim you presented at the beginning. One of the normative theories for is the idea of optimal coding, of trying to preserve information about the stimulus as well as you can, and encode it in the neural population as well as you can. And so if you've done a good job of that, you should be able to decode it, and you should be able to reconstruct the input. Indeed, that's how Olshausen and Field and all those were trained. Now, we asked, can we, from the fly visual system, reconstruct its input?
Yeah.
And if we use that as an objective function, if we found the model that's able to do that well, will that correctly predict the tuning properties, the response properties across the network? And the answer is no. And no
to what? No to
that objective function does not correctly predict the tuning properties of individual neurons within the circuit.
Yes, but let me jump in. I mean, we know from machine learning that perhaps a better architecture would be kind of auto encoding, where you need a bottleneck in the middle to actually generate a nice representation.
Well, let me just. I'll tell you what we did. We took the top half of the network.
Okay, we are getting it. Right? Okay.
Half of it, and we asked, can we decode the input?
Okay.
And so, that should be it. This is an over-complete representation, right? There's 8 photoreceptors per ommatidium, per pixel. And, we have way more cell types and neurons for those. So in principle, you should be able to decode. And and you can ask, is this an optimal, is this system doing optimal coding?
But I didn't understand what you observed so far is that it's not the network is not able to reconstruct, or that the representation that results from reconstruction is not optimal, or doesn't correspond to the data.
It's the latter. The latter.
Oh, it's the latter.
Whether you I should go back and look, but I think it also didn't reconstruct very well. So I think both are true, but certainly the latter I'm very confident about. And to me, this system has to do a lot with very little, and this is one step on the way from perception to action. And action doesn't require that you encode everything about the stimulus. And so, I'm just curious about where we are as a community in thinking about autoencoding or optimal coding as the dominant principle for perception versus more task-driven representations, which need not be optimal coders. And have we moved away from it enough yet?
But I think I think that goes back to what SueYeon showed in one of her slides, that I think the accepted notion is that some early stages are dealing with optimal coding in the sense of information about optimal coding of the information about the stimuli, where downstream, and the question is, where is it downstream? In the case of vision in the mammalian brain, downstream may be high level of the sensory or the perceptual system. In the flight, downstream may be closer to the action. Will be something which is optimal coding per ensemble of tasks rather than storing the information. But I think that's one of the key questions, open questions for us as a community, in really extending the early theories of efficient coding for early stages to larger scale in the brain. That's essentially what we have to do, because we have we're getting more and more information about the data, the behavior, large-scale recordings of activities of large regions in the brain, and connect them. So I agree with you. This is one of the fundamental challenges. Absolutely right.
Yeah, I mean, just to be controversial, I'll just say that maybe optimal coding was never the right framework, and that even task-optimized networks have to cover the stimulus space in their first layers. And what we were seeing was just a consequence of that the first layers of the network just have to, cover the stimulus space. And so, that's why, and the idea that the optimal coding networks also converged on the same codes was just by chance.
Could be. It could well be, because we know that despite the fact that that what I mentioned as one of the success stories of efficient coding is the Gabor filters, and we know that subsequent work showed that there are many other ways
of getting
it. Sparse and kind of non-linearity, non-Gaussianity, and so on and so forth. So you may be right that the essential principle will be something which has to do with the relevant computations that are needed to be done downstream. So we will see. I think that's the kind of questions that SueYeon and you and all of us that are interested in this in sensory coding are faced with. Okay, we have a few minutes left. I would like to ask one question for you, SueYeon, and one for Srini. SueYeon, we talked about possible extension of the geometry of manifold to perhaps to continuous estimation and so on, as Srini brought it up. We can also go to the other direction. Can you extend it to more complex cognitive structures, perhaps downstream of IT cortex or multimodal? How do we build on these geometric principles higher level cognition? Do you have any speculation about that?
Yeah, I didn't have time to talk about it in the talk, but there are many extensions to more cognitive computations, for instance, context-dependent categorization. So technically here, it could be something like manifold capacity, but when you introduce context dependence as a nonlinearity in the readout, but that can be more related to the context-dependent decision-making in like relevant for PFC or more cognitive brain areas. And I think so if once you get those figured out, we have some early work on that domain. And then once you nail down the geometry underlying this sort of more nonlinear computations, although by focusing on piecewise linearity, because it's easier to do theory in that space, then you can start to tackle more complex computations. In my view, but I think more broadly this paradigm or the analytical technique is pretty generalizable to many computations by turning it into the piecewise linear problems and then introducing this statistics of the points that matter from the perspective of the decoder, such as worst points or what we call anchor points. Yeah.
But the piecewise linear is more extending it to maybe more complex decoder, if I understand correctly, or classifier. But I was actually referring to more complex cognitive task involving multiple objects moving. A more complex, still a cognitive task which need to have a notion of objects underlying them, but right. It's not enough. It's not enough to say this is a cat and this is a dog.
Right.
But now what they're doing to each other, actions and how they manipulate the things and so on.
Right.
Do you see a way to kind of extend in this direction?
Yeah. So I think it's possible, although it needs serious work. By extending it to the decoder with multiple outputs, for example, then we can start to go into dealing with multiple objects or multiple latent variables. And how do you encode multiple latent variables in a simultaneous manner? And I'm pretty optimistic.
Okay. It will be exciting to see how this unfolds. I would like to ask you, Srini, you mentioned already the challenge to gain some kind of understanding or principles, from these detailed connectomics and detailed simulations that you showed us. But maybe a different, slightly the same question, but put it in different angle. And this is the question of the role of abstract or simple models. So we have been using traditionally in the field for 40 years simplified models, and there's also been the tension between two camps. One said, well, we need all the details, and other things say, no, the principles are sufficient for the principle sufficient to look at the simpler model, but maybe you have tools, both the data and your computational methods, to actually do perhaps more systematic work in illuminating basically bridging the gap in some sense between simple models. What does it mean simple? How simple? And the full detail models. Can you comment on that?
Yeah, no, I think that's a great question, and it's fascinating. It's something I grapple with a lot because we build complicated models, and I'm of course inspired by the work that you and others have done in building simple models. And how do we get from here to there? And can we even get there? Maybe there won't be a simple understanding to the kinds of things that are possible. But of course, we have to try to build up our own intuition. Like when I think of the Hodgkin-Huxley model of an action potential, that model, of course, can generate a rich variety of behavior. But when it was taught to me by my advisor, Sebastian Sung, in his course, he showed a couple of different regimes. He said, here's one thing that can happen, and here's another thing. Like there's an after hyperpolarization, which can, the post-inhibitory rebound is something where, inhibition can generate an action potential. So, there's a few vignettes that we take away from the complexity, because, the full complexity is, of course, hard. So, the two ways that I can imagine us going about this, one is, very formulaic. It's sort of like there's this framework of model order reduction. So you can say, here's simple dynamical systems that we know what they are. There's attractors, there's oscillators, there's, all of these things. Can we look for them in an automated way within the circuit? And if we do, then we can say, oh yeah, there's, here's an attractor, here's here's a basin that forms and is pulling the system towards it, but then it goes away, and the kinds of dynamical pictures that we talk about in classic systems neuroscience. That we can do. I think we can automate that. I think we can, some of the techniques developed by Laura, with David Tassilo, who've been looking at deep neural networks and then reverse engineering from these dynamical systems perspectives. We can use those, but I think we should also probably look for sort of case studies, like what certainly we want to look for the hydrogen atom. That something simple that we can explain, a minimal circuit that does something, and so we can create those. So we can ask, here's this full network, here's the task that we're trying to perform. How many of these neurons can we take away? And the performance is, this task performance is basically the same. And so, for individual computations, we can try to pull out the minimal circuits. Also, we can do that automated. And the third might be, a couple of worked out examples of here's how the fly does X, or here's how the system does Y. Might be another way that we derive intuition, and we say, oh yeah, we know how this is done, we know how that's done, then maybe, somewhere in between is what just stepping back, I'm trying to characterize what understanding even means.
Sure.
And I don't have that yet.
Sure.
But it's been forced upon me to sort of think about, and so these are three kinds of understandings that I've come to sort of characterize. But I'd love to hear more about different ways in which to grapple with complexity.
Yeah. Okay, so I think I'm taking in your fantastic work, and really, I think both of your work are showing what the future of computational neuroscience will looks like for the next decade or beyond, with new ideas, new data, new tools, and big questions. Some of them old, some of them new. And I think that's the way the field will remain very exciting, very challenging, and with a lot of opportunities for great discoveries of the principles of how the brain takes in stimuli and generates cognition behavior. Thank you, both of you, and I think we'll close this session with this.
You.
Yeah.
There's the applause.